everybody! You are listening to Through Time and Clades. My name is Joan,
And I'm Albert.
and we are back with another Nature News episode. This time we're going to be covering science stories from August of 2024. Before we jump into that, how have you been, buddy?
I would say I've been pretty good, but it's definitely getting to a very busy time of the year for me. Yeah, it's just been a bit of a mad scramble, really, and I know that's gonna be the case for at least a month or two. But that hasn't stopped me from keeping up with the nature news, and so I'm definitely interested in sharing this episode's stories with you all. How about you?
Oh, absolutely. Well, I've certainly been keeping busy too, but probably for different
Hmm.
reasons. So we're currently recording in September, uh, and as of last week, I turned 30 years old.
Oh wow!
So I'm
Congratulations.
finally, thank you, finally in the 30s. And boy, am I feeling it. Because, if you had asked me, when I turned 28 or 25, how do you feel on your birthday? I would say I don't really feel that much different.
Hmm.
But now I definitely cannot say that because my bones and my joints are starting to get the early signs of
Oh,
of old ladyism, and I'm I'm gonna have fun dealing with that. But that's as long as I keep my exercise and I don't push myself too much, everything should be fine. Just take precautions, that sort of thing. Um, and so, I mean, I've been kind of taking the whole month to just celebrate it. It's a big milestone. Lots of hanging out with friends and family, driving over to see my sister, and going shopping and all kinds of stuff. So it's been very nice.
hmm.
It's been absolutely nice. I've also been kind of having fun with David Attenborough documentaries.
Ooh.
So I've been kind of doing this fun marathon where I started watching Life on Earth again because I think it's great. And then I was like, well, do I just go into the Living Planet right after that? Or do I take this opportunity to catch up on some things? And so I decided to have it where I'd watch an old series, then a new series, then go back to old, then go back to new, and so on. So I've been able finally to watch and catch up on a lot of the recent BBC landmark series that David Attenborough had narrated or hosted. So I finally seen Wild Isles. I finally seen The Mating Game. I'm currently working through Mammals,
Hmm.
which I'm also enjoying. I'm not quite done with that yet as of the time of recording. Um, and it's interesting. It's interesting doing it that way. And I know we've talked about similar things before, but the format and style of documentary filmmaking has definitely changed over the decades. And to go from something like The Living Planet to, say, The Mating Game, it's apples and orange. They're almost two completely different experiences. The only thing that is really shared between them is, of course, Attenborough and the fact that they are featuring wildlife and wildlife footage that was the best of the time. But everything from the formatting to the technology to the information we have about these animals and plants has all changed, and it's been really enjoyable to kind of look through the decades in this way. And I'm hoping to keep it up at least for a little bit longer, catch up on some older shows I haven't seen in a while, and as I anticipate, you know, Hidden Planet and Asia and all the new upcoming shows that
Hmm.
we're going to look forward to. We finally got the confirmation, a little bit of follow up, that Attenborough will be hosting Asia. They released some more information on that, some more hints at scenes that are going to be shown. So, again, fingers crossed. If they're going to commit to a 2024 release date, we might get a teaser here within the next few months. We can only see. But no, otherwise, I've been doing very well. It's been a fun month, and I'm definitely excited to share some of these stories as well. It was another big month of stories in August, and so to kind of weed through some of the ones we wanted to tell while not, repeating a lot of similar topics and themes, I think we found a pretty good medium. But before we jump into our news stories, I did have a mini review that I'd like to share. Do
Hmm.
you want to go ahead and jump into that?
Sure thing.
Absolutely. Let's hop to the next slide now. So this book that I want to talk about today is called "We Are the Middle of Forever: Indigenous Voices from Turtle Island, on the Changing Earth, which is a collection of 20 mainly transcripted interviews with North American indigenous individuals who have been actively working in the fight against the worst effects of the Anthropocene event. Now, these were gathered and edited by journalist Dar Jamal, who is known for his previous work covering the U.S. invasion of Iraq, and Stan Rushworth, who is instructor emeritus of Native American literature and critical thinking surrounding indigenous issues at Cabrillo College in Aptos, California, who sat down in person or over Zoom to conduct the interviews, because it's very clear throughout the book that a lot of these took place during COVID time, which was a pervasive topic throughout. Uh, the authors were concerned about current responses to the Anthropocene event, current mainstream responses, if you will. And they wish to seek out various indigenous perspectives on these issues, given that Native Americans have faced their own rapid, dynamic environmental and cultural changes as a result of European colonialism for over 500 years and have still remained adaptable and resilient. Their experience should be enlightening. And indeed, as I read through each interview, I was certainly enlightened. This is one of those books that a reader must take their time with and reflect on after each essay has been read, which is what I ended up doing. Subject material in each varies from person to person. So some of them were going more into the interviewee's personal experiences, and others going more into understanding overconsumption or land management or indigenous representation in US politics. Some of the discussions I found especially fascinating and moving concerned indigenous philosophies and the ways that different nations have interacted and continue to interact with each other and with their surroundings. And I say that because it's clear that making distinctions between humanity and nature is unwise and has led to many of the thought processes and beliefs that got us to where we are today with such dramatic environmental issues. And I also appreciated the values and promotion of community and social networks and the encouragement of conversations about past traumas and tragedies so that proper healing can begin. Even something as seemingly simple as taking the time to stop and listen to your surroundings, literally reconnecting with the earth, I thought is a profound and educational experience. And so I decided to make this a mini review, unlike some of my other book reviews, simply because I feel very strongly that the voices in this book need to be heard for
Mm.
themselves. Rather than summarizing and going chapter by chapter like I did with, say, The Dawn of Everything or The Memoirs of Cleopatra. I want to encourage readers and potential readers to take the time for themselves to engage with each interview. And then, if they wish to engage even further with the topics concerned, go to the back of the book where they have given a series of questions uh, for each interview. So that way you can do some philosophy of your own and think about what positive impacts you can bring to shaving the severity of the Anthropocene events and bring about a better world for us all. And so I immensely enjoy reading this book. I think I got through it in a week and a half, which is a pretty good sign that
Mm.
I really enjoyed it and was taking a lot from it for my own personal use. And wait, things that I'd want to share with others as well to consider. And so I really appreciated reading this, and I definitely, give a big shout out to Dar Jamal and Stan Rushworth and all the interviewees concerned for putting together a very special book. So that's really like what I wanted to say about this. What do you think, buddy?
I mean, I haven't read it, but that sounds like a really interesting and thought provoking book. And obviously, it promotes some frequently underrepresented voices. So, based on what you've described, it certainly sounds like a book very much worth reading.
Absolutely, I couldn't agree more. And so we will definitely put links to purchase the book in the description below, as well as some other additional information about the authors, and that way you can go and explore for yourselves. We highly recommend it for sure. This is a Through Time and Clade's 11 out of 10 if we were to do ranking systems on our show. Um, so that's it for my mini review. now we can go ahead and jump into our news stories. This is a more traditional news episode this time around, so we both have two stories each that we'd like to share. And Albert, I believe you have first dibs.
Hmm,
Would you like to go ahead?
sure thing. Alrighty, so our first story is going to be about freshwater fishes, which I think are a set of animals that don't always get the attention that they deserve, I suppose. If you think about, say, I mean, we were just talking about nature documentaries, so if you think about fishes in nature documentaries, I feel like a lot of the time they're mostly going to be marine fishes, especially fishes on coral reefs or big predatory fishes like large sharks. And sometimes maybe deep sea fishes if the documentary has the budget to go down to the deep sea and do some filming there. But yeah, you don't really see the freshwater fishes as often. And that's a pretty big shame because, interestingly, even though fresh water takes up a lot less of the Earth's surface area than the ocean, over half of all living fish species, by which I mean non tetrapod vertebrates, are found in freshwater ecosystems and not the ocean. So think about that. That sounds very counterintuitive, not what you would expect based on their representation in popular media, probably. But yeah, there are more species of fish living in rivers and lakes and ponds on Earth today than in the ocean, which is a much, much bigger expanse of water. In fact, I think in terms of those freshwater ecosystems that we mentioned, they only account for about 0.01% of all the water on Earth. So, yeah, it's a very, very disproportionate representation of fish diversity. But it kind of goes to show like how diverse and how complex these freshwater ecosystems can be. Freshwater ecosystems are quite fascinating today, but it seems that something quite interesting about them in the distant past is that they appear to have been one of the main ecosystems that were relatively unaffected by the end-Cretaceous mass extinction, that most famous of the major mass extinction events. So, of course, this is the mass extinction that wiped out most of the dinosaurs, other than birds, many, many different types of animals on land and in the oceans. And yet, as far as we can tell, freshwater ecosystems appear to have been a lot less affected by the end Cretaceous mass extinction. And to an extent, this makes some sense because we think that one of the main killing agents, as we say, during the mass extinction at the end of the Cretaceous was this gigantic asteroid that impacted the Earth. And of course, when it impacted the Earth, it caused massive destruction to its immediate surroundings at the site of the impact. But probably what really caused things to go extinct all over the world was the debris that impact threw up into the atmosphere. And that debris, well, it did several things. One was that some of the heavier particles rained down from the atmosphere, generating heat and destroying pretty much anything that hot debris landed on, including green vegetation. And the lighter particles probably stayed up in the atmosphere and blocked out the sunlight, which would have killed off any remaining photosynthesizing organisms out there. And so, with most of the green vegetation gone, that would have disrupted food chains and food webs all over the world and probably killed off many of the animals that depended on those green food sources. But freshwater ecosystems are a little different from your typical ecosystem on land or even in the ocean. Because freshwater ecosystems, the base of their food chains does not depend so much directly on green vegetation. Instead, a lot of the base of the food chain in freshwater ecosystems is formed by detritus that gets washed down into the water from land. And so that's a kind of resource that would not have been immediately depleted by the destruction of green vegetation. And so it may have been that freshwater ecosystems still had kind of enough to kind of buffer them through this period when all the other ecosystems were suffering. And furthermore, animals that were living under the water, like in freshwater ecosystems, would have also been buffered from the kind of raining hot debris that was falling down from the sky. And so freshwater kind of organisms were in a pretty good position to survive the worst of the effects of the end-Cretaceous mass extinction. This seems to have been a pretty consistent pattern. So, for example, in groups like turtles or crocodilians, we do seem to see this pattern where the freshwater members of these lineages, in many cases, appear to have made it through the extinction event relatively unscathed. However, one very important group of freshwater animals, the fishes, largely have not been studied in this manner, so we don't know very much exactly about how freshwater fishes were affected or not by the end-Cretaceous mass extinction. And there are some reasons for this, and one is that the fossils of freshwater fishes tend to be pretty incomplete most of the time. And you can see some examples of freshwater fish fossils on the slide here, and you can see that they're basically isolated bits of vertebrae, and they're very tiny vertebrae at that, for most part. And so they're not that easy to study because, in fact, in many cases, it's very difficult for us to determine even what kind of fishes these bones belong to. And partly this is due to these fossils being understudied in general. So we don't have a very complete understanding of how to identify freshwater fishes by isolated bones alone. But another aspect of it is the fact that it's so incomplete. I mean, if we did have more complete fossils, we might be able to tell what these fishes were, but we don't. So, but the authors of a new paper that came out in August have tried to approach this problem through a couple of different ways. The first thing they did was that they looked at phylogenetic trees, so tree diagrams documenting the evolutionary relationships among different groups of organisms. They looked at phylogenetic trees from genetic studies of modern fishes, and specifically freshwater fishes, and then used these phylogenies to try and estimate the rates of freshwater fish diversification over time to see whether any major diversification events were kind of correlated with the end of the Cretaceous. The second thing they did was they looked at the fossils. So they kind of looked at specifically fish fossils from a site in the Denver Basin of Colorado that spans just before to just after the Cretaceous Paleogene boundary. And so this is a very, very valuable site because it shows us exactly what's going on, like just before and just after the extinction happened. The fossils that were found like just before and just after are estimated to have been only 185,000 years apart, which of course is a very long time from a human perspective, but is very, very short in geologic time. And so from these two different angles, from the perspective of molecular phylogenetics and from the perspective of fossils, the authors wanted to see if they could detect any kind of pattern regarding freshwater fish diversity across the end Cretaceous. And so their diversification analyses using molecular phylogenies, they found evidence that there was increased diversification within a few groups of fishes shortly after the end Cretaceous mass extinction. And so these groups include a group within the Characidae. So this is a group that includes tetras. If you are familiar with like fishes that you commonly keep in aquariums, the tetras are a very commonly kept and diverse group and often very colorful group of freshwater fishes. And also two clades within the Cypriniformes also appear to have diversified intensely shortly after the end Cretaceous mass extinction. And so Cypriniformes is a group that includes things like carp, including the domesticated goldfish, and apparently there are like two major groups within the Cypriniformes that show evidence of like extreme diversification afterward. But But for most part, there doesn't seem to have been much strong evidence for many other major diversification events in freshwater fishes after the end Cretaceous, at least based on these molecular phylogenies. And to be fair, the authors do acknowledge that there are some limitations to this approach because these molecular phylogenies, they're based on genetics, which we can only get from modern or these very recently extinct taxa, and so they don't include things like fossils or fossil members of these major lineages. And so it's quite possible that there were other diversification events that happened, but just were not recorded in these molecular phylogenies because maybe most of the descendants of these diversification events ended up dying out later, and we just don't have their genetics to work with anymore. But nonetheless, at least based on the evidence that we do have, it does seem that there were some diversification events in freshwater fishes that may have been triggered by the end Cretaceous, but not that many. And what do the fossils themselves say? So, like I mentioned earlier, it's very hard for us to identify specific groups of fishes based on fossils. So, what the authors instead focused on were not trying to identify like what species or what major group these fossils belong to, but instead sorted them into what we call morphotypes. So, at least like identifiably distinct categories of fossils based on their morphology. So you can say this one looks different from this other one, even though they're supposed to be the same bone in the body. And of course, this does not necessarily correlate with the number of species that were around, but it does serve as a pretty rough proxy. And so, of course, there are species that can look very similar to each other, and so you might have two different species that have very similar morphotypes. Or you can have like one species where there's a lot of variation, like between adults and juveniles, or between like different sexes, and so you might have like two morphotypes representing the same species. But nonetheless, going by morphotype still gives us a rough idea of like how diverse fishes were, like before and after the end Cretaceous mass extinction, at least at this one location. And what they found was that there were some changes across the end Cretaceous boundary. So there were two fossil morphotypes that were already relatively rare that were only found before the end Cretaceous boundary and not afterward. Whereas two other morphotypes were found after the boundary but decreased greatly in abundance. And interestingly, there was one morphotype that was very rare before the boundary but became the most abundant morphotype afterward. And so you do see some localized effects, changes in abundance and diversity of freshwater fishes, at least when you look from a morphotype perspective across this boundary in this one location. But nonetheless, there isn't much evidence for a lot of major extinctions going on within the freshwater fishes. Like only two fossil morphotypes were lost in this assemblage, and they were already rare to begin with. And so it is possible that they survived, but it's just that there weren't fossils found afterward. And so overall, these results kind of from both the molecular phylogenetic perspective and from the fossil perspective do support the idea that freshwater fishes were less affected by the mass extinction than many other animal groups were. But they also provide evidence of localized impact. So there were some changes in abundance and diversity in this region, at least, and the authors do mention that we can't really extrapolate these patterns to say that this was what was going on all over the world. But it does show that the end Cretaceous mass extinction did have some effects on freshwater fish diversity and abundance. So it gives us a more complete and nuanced picture of what was going on with freshwater ecosystems during this pivotal time in Earth history, while also kind of supporting our general concept that freshwater ecosystems managed to make it through the extinction relatively well compared to most other types of ecosystems. And so that is what I have to say for this study. Do you have anything to add?
Well, I think it's fascinating to consider the relative survivability of freshwater ecosystems across such a, horrific and
Yeah.
devastating mass extinction event. Um, it really brings to my mind discussions about, our current mass extinction
Mm-hmm.
event, or
Yep.
at least like the beginnings of our current mass extinction event, where you'll sometimes hear it mentioned that, this is the worst rate of extinctions and wildlife losses since, the asteroid impact, the KPG. But considering, that freshwater ecosystems seem to have made it out, relatively unaffected, as you say, through that event, versus nowadays where freshwater ecosystems are some of the most at risk ecosystems,
Mm-hmm.
due to the environmental impact. It's like, yeah, like in that sense, it's probably a lot worse than the KPG in that respect. Um, which that's a whole other can of worms we could go into.
Mm-hmm.
Um, but it's very fascinating to see a study like this. Interesting to consider like this one region had so many different variables going into it. You had groups diversifying after the impact. You had groups that were decreasing in number, and others that were just kind of biding their time and then took the opportunity to diversify afterward. I am curious to see whether that impact or that rate is the same across other um, freshwater sample sites that we might have across the
Mm-hmm.
world. I know our range of sites for like the very end of the Cretaceous and into the Paleocene, um, are not that large to begin with.
Right.
Um, at least in terms of like complete ecosystems, like say the Hell Creek Formation. I'd be very curious to know like how we are with just kind of freshwater ecosystems, for example. Um, but it's neat to see. It's neat to see, and it does track with a lot of the studies that we've talked about on the show before. For example, the fact that we understand that many groups of reptiles, also like freshwater reptiles, also made it through, or seem to have made it through relatively unaffected, and even retained sizable body proportions
Mm-hmm.
and scales through the event. Where, and not long after the KPG, we're having giant turtles and giant, crocodilomorphs roaming around. Um, so it's not surprising, but it's also good to know that freshwater fish seem to have undergone their own kind of safety across the KPG with some minor changes. Because even like mentioning like the reptile, like freshwater reptiles going through this, there were still key losses. There
Mm-hmm.
were lineages that were lost and others that were reduced and would eventually also die out. The campsosaurs come to my mind,
Mm-hmm.
where we had this lineage of diapsid reptiles that came in different body types. The Champsosaurus is the most familiar one. It's very crocodilian-esque in body form, but as I understand it, they are not close relatives of uh, crocodilomorphs at all, and they survived the KPG. But it was like by the late Eocene or early Oligocene is when they went extinct. So like they had their time, and then uh, they lost out.
Mm-hmm.
And so now the only lengthy crocodile-like animals in the waters that we have left are crocodiles and
Mm-hmm.
alligators, and their kin. So yeah, a fascinating paper. I'd love to see follow-ups on this for sure in the future. Now, speaking of diversification across time, my next story concerns that very similar topic as well. Shall we move on?
Yeah, let's do it.
All right. So on the next slide, my first paper comes to us from Jamie B. Thompson and colleagues from the journal Nature. Long known as symbols of the desert, cacti are in fact a mainly American clade of plants, the Cactaceae, which can be found also in wet, cool, and forested environments. They're not desert specialists specifically. This clade belongs to a larger group of plants called Creophyllales, which are characterized by, of course, similarities in DNA, as well as simple xylem vessels. So these are the cell tissues used in transporting water from the roots to the rest of the plant, as well as by the possession of beta-lane pigments by most members of this group, which gives the tissues a strong reddish color. Thus, cacti belong to the same wildly diverse group of plants as carnations, beets, and other types of plants called succulents. Now, cacti are succulents too, and that just means that their stems have become extra widened and curvy to allow for extra water storage in hot, dry environments. This has had an effect of transferring the role of photosynthesis from the leaves to the stem. Now, the leaves of a cactus, depending on the species, have either been reduced or lost, as well as developed into their signature feature, their spines. So the spines of a cactus are actually derived from modified branch shoots called areoles, and it is this that distinguishes them from the other succulents in the Creophyllales clade. Now, as anyone who's ever been unfortunately stung by a cactus will tell you, these plants have excellent and effective defenses. It's typically thought that the spines are a plant predator deterrent, but it is also now known that spines can also help with providing shade to the stem. So, in the heat of the desert, having a dense outer covering of spines acts like an umbrella, preventing the fullest extent of sunlight from reaching their tissues and killing the plant. They can even function to reduce airflow across the stem and can help direct rainwater down into the roots, especially if the cactus has a ribbed structure around. Now, the spines of some species can also produce sugar glands that attract insects like ants to their flowers. Things like the genera Cactus and Coryphantha. Cacti have been known to attract pollinators to their often vibrant flowers, organisms as diverse as moths, birds, and bats. In all, there are over 120 genera and 1,700 species of cacti, ranging from the Caribbean Leuenbergia, which seems to be the oldest diverging lineage, and one of the few genera with full leaves, to the tiny members of the genus Epinthalantha, which are only a few centimeters wide, to the famous saguaros of the American Southwest,
Hmm.
which is the largest species, averaging to over 12 meters high, with the largest ever having measured 23.8 meters tall. Now, the saguaro Carnegie gigantea, yes, named after Andrew Carnegie, also takes distinction as being the cactus with the longest known lifespan, it being recorded that the individual called Old Granddaddy was around 300 years old when it had begun to die of bacterial necrosis in 1992. There are other curiosities to the cactus kingdom, like the mistletoe cactus, Rhipsalis bactifera, which is the only species to naturally occur outside the Americas, growing in Central Africa and Sri Lanka, where it must have been dispersed naturally, somehow. It's been proposed maybe birds dropped their seeds, or maybe they floated over the oceans. This is an area of debate.
Hmm.
Or how about the jumping cholla, Cylindra puncia fulgida, which can increase its range by having special spine segments that stick to the hides of passing animals with a sharp sting, kind of like a wasp sting. And that causes the animal to run away a short distance before kicking them off, where the spines can take root into the soil and grow into a new plant. And then, of course, there's the infamous peyote, Lophophora williamsii, which, if dating techniques are anything to go by, was used by southwestern indigenous groups for at least 5,700 years. One of the few instances of psychoactive substances that has been verified in pre contact American societies. All this said, it has been understood by evolutionary biologists that, as far as succulents go, cacti have among the fastest rates of diversification, even accounting for their slow lifespans. Now, despite the relative lack of a fossil record, genetics points to an origin for cacti sometime during the late Eocene, by 35 million years ago. And most of cactus diversification took place only within a period of 5 million years during the Eocene, between 10 and 5 million years ago specifically. This is starting to sound like the origin of orchids that we discussed back in our February 2024 news episode, isn't it? In fact, the study which recovered that result by Monica Arakaki and colleagues back in 2011 also found that cacti share with other desert plants like agaves and ice plants a contemporaneous recent radiation. Now, one would think that the aridification of environments leading to the Pleistocene Ice Age would be enough to explain the sudden burst of cacti, but researchers do not seem to be convinced, for it's been noticed that much of this recent cactus evolution has been more concerned with changes in growth and reproductive strategies than with adaptation to hot desert climates. So, what really seems to have driven this change? Rather than analyze one or two evolutionary drivers on a lineage of organisms, Thompson and colleagues decided to analyze several of them, many of them. Pertaining to cacti, the team singled out several proposed drivers from botanical literature over the years. 39 of these potential drivers had been selected, ranging from air temperatures during the day and night and throughout the seasons and the years, precipitation through the seasons of the year, soil and sand content, biomes and elevation, pollination, chromosome counts, growth forms, etc., etc. Studying so many drivers is difficult and full of changes now, including the fact that different variables can and do interact with each other. So the team looked to machine learning to help answer their questions. So they constructed a phylogeny, including 1,063 species of cacti, that's over half of all known species, all tied to a huge data set where each of the 39 drivers was accounted for. Now, the machine learning software XKB Boost was used for assessing and modeling the drivers and their interactions, testing models of speciation and extinction to pinpoint the relative importance of each driver for the clades of cacti studied. Now, the phylogeny at the top left showcases their results. That color gradient from blue to white to red represents BAMM, or Bayes factors 4096, which simply put measures the rates of speciation in sample lineages and how much they vary. The range of colors indicates that cacti have so-called heterogeneous diversification rates, with many lineages undergoing multiple shifts across geologic time. And the team was also able to give a new quantification for the origin of crown cacti. They put it at about 37 million years, which is not far from previous estimates. But the main focus of the paper, however, is concerned with the figure below the phylogeny shown here. Out of all the 39 potential drivers analyzed through the machine learning process, Thompson and colleagues recovered two lists of factors in order of relative importance. In list A on the left, XK boost, or XG boost, excuse me, accounted for complex interactions between drivers and found that five of these were prominent predictors of speciation rates with a high prediction accuracy. That's mean diurnal air temperature range, so the mean air temperature during the day, soil sand content, plant size, isothermality, and geographic range size. In list B, XG Boost used a simpler model that removed the complex interactions and found that biome, seasonal precipitation, and chromosome count were more important, at the expense of isothermality and geographic range. Taken together, the team singled out diurnal air temperature range, soil sand content, and plant size as the most important of the main drivers of cactus diversity overall. So, what does this mean? The macroevolution of cacti, that is, the evolutionary change across groups of organisms as opposed to changes within species, was facilitated by a complex array of factors. Regarding the effects of daytime temperatures, it has been known that cacti, hardy as they've been claimed, are actually very sensitive to extreme temperatures. If desert environments are too hot and too dry, that can actually kill cacti, which thus negatively affects their survival in such extremes and even limits their distribution. Thus, many species of cacti have evolved ways to cope in deserts with high daytime temperatures within the ranges that they can tolerate. Think about the shading caused by the spines, for example. For soil sand content, botanists and plant growers alike understand well that cacti, with their shallow roots, do best in sandy soils that do not hold too much water and have a particular mix of organic and inorganic materials. And there is room for variation in this between different species. Thus, cacti have evolved to become ecologically specialized to particular soils across their mainly American range. Hence, how some species can even grow in tropical forests or cooler mountain ranges. And plant size affects cacti from the smallest to the largest, bringing in adaptations for longer lifespans and unique growth patterns that encourage survival. So, all of these factors seem to have worked together to give us the wide diversity of cacti we see today, and give us important insights and further areas of study to examine these specifics that have led us to the 1,700 species alive today,
Hmm.
which are, of course, currently threatened with now disastrous environmental changes that will make things even warmer for their already desert adapted environments. And so it was really neat to take some time to look at plant evolution and even for organisms as familiar as cacti, there were still surprises in store for them as well. So I really enjoyed covering this paper. Do you have any thoughts, Albert?
Yeah, I mean, this sounds like a really interesting study. And certainly, I would say like cactuses are among my favorite types of plants. They do have that kind of endearing quality to them. And I've kept a few as houseplants before. And I've also run into them during field work when I was studying as a geology student. Fortunately, not literally ran into, but I have run afoul of their spines on a couple of occasions, and they are effective, as you say, for deterring potential predators. One of my favorite fruits is a dragon fruit, which is the fruit of a cactus. But I definitely did not know a lot of the background about their evolutionary history or diversification. I'm happy that there are people like out there. There are researchers out there studying this very fascinating topic.
Absolutely. And now I understand you yourself have another plant paper to share with us, this time more broadly looking at the flowering plants.
Yeah, let's go to that one. Um, so yes, it is funny. We both picked studies related to diversification and specifically diversification rates of flowering plants. And so, as you mentioned, this one tackles the topic a lot more broadly. So we're going to talk about the diversification of all flowering plants in general, and not just cactuses, and specifically when flowering plants originated, because that has been one of the absolute biggest controversies, I would say, in plant evolutionary studies, or at least like macroevolutionary studies. So the oldest fossils that we have currently found of flowering plants date to the early Cretaceous, so around 130 million years ago, a bit more than that perhaps. And these fossils are fossils of their pollen. And you might not think about this, but pollen is actually an incredibly valuable resource for paleontologists. It's made of some of the toughest materials that any organism can produce. So pollen is something that actually preserves really well in the fossil record. Now, of course, pollen is really small, so when you study pollen fossils, you do have to do it under a microscope. And the pollen of different groups of plants can also be very distinctive, so you can easily identify what groups of plants were around at the time. And of course, if you know what plants are around, that also tells you about something. That also tells you something about what the climate was like and what the overall environment was like. And so, the pollen fossil record is in many ways one of our kind of best windows into the evolutionary history of plants. And the oldest fossils of pollen that we can definitively identify as belonging to flowering plants come from basically the beginning of the early Cretaceous. And a little bit later than that, around 125 million years ago, also early Cretaceous, we have the earliest known kind of what we call macrofossils of flowering plants. So fossils that you don't need a microscope to see, basically. So these are large plant parts like leaves and stems and such. But we know that the fossil record is incomplete. And so the oldest known fossils that you find of a given group are almost certainly, just statistically speaking, not going to be the absolute oldest members of that group that ever existed. And so to try and figure out exactly when groups evolved, one of the ways that we approach this is called using what we call molecular clocks. And molecular clocks, what they do, we have discussed this before on the show, but just to briefly summarize, they compare the genetic or protein sequences of different organisms and then try to estimate how long it would take to accumulate all those differences between these different organisms. Now, of course, you can only use the molecular clock method on things that we have molecular sequences for, and so this mostly means groups for which we have genetic or protein data based on living members or somewhat recently extinct members. But in any case, there are obviously a lot of flowering plants alive today, so people have tried to use the molecular clock method to try and infer when flowering plants originated. And the results of molecular clock analyses when it comes to flowering plants are pretty curious, because some of them place the origin of flowering plants in the late Jurassic, which would not be too far-fetched, but others place them much older than that. So maybe in the early Jurassic, or even the Triassic, or even the Permian before the dinosaurs were around. And this is, well, first of all, that's a very broad range of dates. It would be nice if we could narrow that down a bit. But also, it's like, wow, that's much, much older than the oldest fossils that we have of them. We're talking about like a discrepancy of, broadly speaking, 50 to over 100 million years older. What is going on here, right? Um, I mean, yeah, the fossil record can be incomplete, and so it is not that far fetched to suggest that the origin date of flowering plants was a bit older, or somewhat older, than the oldest known fossils. But is it really so old that we haven't found any identifiable fossils of flowering plants in like the time span of 50 to 100 million years in the rock record? Um, that is something that seems, at least a bit more questionable, at least a bit more up for debate. And to this day, there is still no, I guess, major consensus about exactly what the solution to this problem is. And so the authors of a new paper review this controversy, and this is an interesting paper because it is what is called a viewpoint paper or perspective paper. So it's not really a paper that's dedicated to doing like a detailed analytical study of this topic. And it is also not quite like a comprehensive review paper where they go through like all the different stuff that has been said about this topic ever. But it does a little bit of both of those things basically, and it's basically an outlet for the authors to kind of discuss this topic, and kind of like maybe add a little bit of their own perspective into this. So they do briefly review the controversy, and they do run some of their own analyses that are relevant to this question, but maybe not as in-depth as you would expect for a detailed analytical study or a very detailed review paper. But this does make it a very straightforward paper to read, and so if you want to get a sense of where we're at regarding this topic in a way that doesn't involve too many details or jargon, I think this is a very nice paper to go ahead and read. And so in terms of like adding their own perspective onto this topic, what the authors did was that they say, okay, let's assume then that the fossil record is correct and angiosperms or flowering plants did not originate that much older than the early Cretaceous. So assuming that modern angiosperms did originate like shortly before the early Cretaceous, so in this case they set it at 150 million years ago in the late Jurassic, what would be the accuracy of our molecular clock methods if this was the case? And so they ran a bunch of different analyses basically simulating what results you would get from molecular clock analyses given an origin date of 150 million years ago for flowering plants. And what they found was pretty interesting. So the thing about molecular clock analyses is that we know that genetic evolution does not proceed at the same rates in different groups or throughout time. Like they change over time and they change in different groups, as you discussed with the cactuses, which like evolve really fast, right? And so you can't just take these molecular sequences and straightforwardly say, okay, we'll assume that this many changes occurred over 1 million years or something, and then kind of infer the origin date from that. You do need to give it some leeway to kind of vary the rates of change over time, and then try to figure out what might be the most likely divergence times based on that. The way you kind of what we call calibrate the molecular clock to give it some actual dates that it can use to inform these rate shifts is by using usually fossils as calibrations. So you tell it, okay, so here's the oldest known fossil that we have of this group. So you have to make this group at least this old, basically. And sometimes we can also specify maximum calibration as well. So what I just described was a minimum calibration. Something is just at least this old. But sometimes people will also use maximum calibration and say, we think that it's very unlikely that this group is older than this. So you have to give the origin date a range between like this minimum date and this maximum date, basically. And so it turns out that when the authors ran these simulations, like assuming that angiosperms are 150 million years old, and they used both minimum and maximum calibrations, what they found was that the molecular clock methods routinely underestimated the origin time of angiosperms, and so that's what these diagrams on this slide actually show. So the top two diagrams were the ones where they basically use both minimum and maximum calibrations with their molecular clock analyses, and you can see the range of dates that they found for these different groups. So they show like the origin date of all seed plants, the origin date of gymnosperms, such as conifers and such, and then also angiosperms on the bottom. The true origin date is kind of represented in the middle at the 0 mark, whereas if the estimated origin dates were too young, they would skew to the right. If they were too old, they would skew to the left. And so if you use both maximum and minimum calibrations, it turns out that these dates tend to be underestimated in this case, and so they get skewed over to the right on these graphs. However, if you use only minimum calibrations, which in fact is like the more common method, because oftentimes we can't really often reliably say that something couldn't have originated past a certain date, right? So it's more common to use only minimum fossil calibrations when running molecular clock analyses. And it turns out when you only use minimum calibrations, the molecular clock analyses wildly overestimated the origin date of angiosperms, as you can see on the bottom half of this figure here. And so you can see the purple kind of range for the angiosperms. They are like skewed way to the left when you use only minimum calibrations. The authors interpret this as evidence that it is quite likely that the older estimates for angiosperm origins are being misled by exactly this effect. Basically, the reason that we're getting such old dates from like the early Jurassic to even the Permian for angiosperms is very likely because our current molecular methods cannot like properly account for like when and where the major shifts in genetic evolution rates happen in flowering plants. And so even if angiosperms only originated shortly before the early Cretaceous, our current methods might not be able to detect that reliably because that's just like a limitation of the current models that we have. And this is quite noteworthy because the authors of this paper actually were some of the people who authored papers in the past arguing for like a Triassic origin of flowering plants and things like that. And so they basically changed their minds over the years and kind of come around to this way of thinking instead. And now, if all of this sounds pretty familiar, then you've probably been listening to quite a bit of our show. Because we have discussed like basically this exact same problem with things like birds, with things like mammals, and now the time frame regarding these groups is a little different. So, for example, with birds and with placental mammals, like a lot of the controversy is about how much they diversified before and after the end-Cretaceous mass extinction. So, do we have like a bunch of these groups before the end-Cretaceous mass extinction during the late Cretaceous, or are they mostly like a Paleocene kind of origin? Whereas with the flowering plants, it's more like early Cretaceous or before early Cretaceous. So a little bit of a different time frame there, but it's a very, very similar kind of problem, and it's very, very likely caused by very similar reasons, where like there are like major shifts in evolutionary rates that our methods cannot really account for at the moment. I think people often are very tempted to blame a lot of these discrepancies on the limitations of the fossil record, and it's true that the fossil record does have its own limitations too. But we definitely do have to acknowledge like these, I think, increasingly compelling evidence that our molecular clock methods have various pitfalls as well. And so, yeah, I think this is a very nice little paper to read regarding this subject. And if you want to know even more about this problem, there's actually an earlier and kind of more detailed review about the problem that was published in 2019 by one of our friends and listeners, paleobotanist Mario Corio. So I'll definitely be sure to link that study in the description as well. Obviously, because of my research on birds, where we deal with a very similar issues, I'm always very interested in reading more about like the angiosperm divergence time problem. So this is a very interesting paper to me. What do you think?
Yeah, it's fascinating. It's really glad to. I'm glad to see that the situation is at least understood better through these methods that we can maybe pinpoint solutions in the future. Um, One thing that I've always been curious about with the whole debate about when I guess crown angiosperms emerged in my readings about the history of life and the different groups of organisms that have come about over the past 4 billion years or so, I've come to understand that there's kind of a handful of gymnosperm-esque plants
Right.
that have been allied to the angiosperms, things like cycadioids or Glossopteris, the very famous fossil that was used to kind of help uncover continental drift back in the day. These being more closely related to the angiosperms than to true gymnosperms that are around today, like conifers and ginkgos. And I understand a lot of those now extinct groups have like Jurassic and Permian origins. Um, do you think that they have anything to say about those plants in regards to this controversy, like whether in this paper or in Corio's paper or in general?
So I don't recall offhand like how much those plants have been mentioned in this debate, but yeah, like that's one of the things you do see like these groups that are closely related to the group of interest, like in this time span where we're supposedly missing this huge fossil record, right? I feel like that's something that shouldn't really be ignored. It's like because it does suggest that if these modern groups were around at these times that some of the molecular clock estimates suggest they were around, then they must have been really rare in comparison to like their otherwise very similar close relatives. And so, is it really that plausible that they could have kind of completely avoided detection for such a long span of time in the rock record? And like technically, yeah, in some cases maybe, but especially with plants, I think you tend to have a pretty good fossil record of them, like I mentioned with the pollen and such. Um,
Mhm.
it is like I think fairly questionable, especially like the older you get with these estimates.
Gotcha. Okay. I mean, they're very good points for sure that you've made. I think a lot about how the earliest fossil evidence for grasses is from titanosaur coprolites, I think in
Yes.
particular. And the researchers who analyzed the pollen grains in the coprolites found that they belong to like very widely different groups of grasses, which then implies that grasses had diversified like already fairly strongly in the late Cretaceous, and yet we don't find fossils of all those different types of grasses for some
Right.
reason.
Right.
Um, yeah, whew, this is a this is like one of like the debates of plant evolution, isn't it? I, I'm definitely very curious to see where this goes in the future. I guess like personal opinion time. Uh, a Cretaceous origin for flowering plants does seem to make sense. Just considering the correlations we have between like the diversity of pollinating insects and extinct bird groups and that sort of thing, which a cause effect sort of thing. It makes sense that at the very least, like there was a very huge initial burst of diversification for angiosperms in the early Cretaceous that may correlate with the origin of the group. But yeah, I guess time will tell where we go from here and what kind of questions we'll be able to answer,
Yeah.
if we can at all. Really great paper. Thank you for sharing that with
Oh, of
with us.
course. Yeah, and in fact, that's one of the things that people have mentioned in regard to this debate is that plants exert such a major kind of influence on the rest of their ecosystem. Right? Like a lot of ecosystems themselves are basically defined by what types of plants are present. And if there really were like a bunch of like hidden angiosperms like all along this time period where we don't find their fossils, and yet some of the molecular clocks suggest they were present. That has a lot of implications as to how we interpret the ecology of the ecosystems that were around at the time. Isn't really evidence one way or another, but it is something we have to think about.
Absolutely. So I think in that case, then we'll shift gears to my last story, and we're going to kind of leave the plant world and go back to the world of animals for this one. Um, so my second paper comes to us from the late Jeffrey L. Winnell and colleagues from the Royal Society Open Science Journal. The phylogeny of snakes has undergone quite a bit of change over the last decade or so, especially thanks to advances from DNA research. We now have a somewhat improved understanding of how these snakes of the world relate to each other, and have found that some proposed groups in the past have needed to be reorganized or even rejected. One such example is the clade Colubridae, which used to encompass the great majority of the so-called advanced snake species that lacked the derived characters of the vipers and elapids. You could say it was the perciforms or insectivora of the snake clade. So it was perhaps no surprise that when DNA studies began to be done on snakes, that it was discovered that this group was paraphyletic with respect to vipers and elapids together. So herpetologists have since split up the group into anywhere from 6 to 18 clades, making up the larger clade Colubriformes. That at least has 5 major subdivisions within. Pariidae, which are the slug snakes of eastern Eurasia, Viperidae, the pit vipers, the adders, and the rattlesnakes, Homolopsidae, which are the mud and mangrove snakes of Southeast Asia and Oceania. Then there's Colubroidea, which is a highly diverse group, including rat snakes, racers, water snakes, and tree snakes. And then the focus of our study today, Elapoidea. Now, this is a huge lineage of over 700 species that's found across the tropical and subtropical regions of the world, both terrestrial and aquatic. All this diversity is split into 9 subclades, of which the Elapidae is probably the most familiar. This group includes some of the most venomous snakes in the world, with neurotoxic venom that destroys nerve tissues, that is injected from non-erectile fangs that are stored in these little special pockets in the flesh of the mandible. It's here where we find the cobras, genus Naja, which is shown at the bottom left, and their relatives, including the king cobra, Ophiophagous hana, all of which can iconically rear themselves up and fold special flaps of skin on their neck. This is the hood as a defensive display. Some species can even shoot their venom at considerable range. Then there are the mambas, like the infamous black mamba, Dendroaspis polylepis, which are widely feared for their fast slithering speed and deadly venom that can kill within minutes. Possible headliner for the world's most venomous snake also belongs to this group, the inland taipan, Oxyuranus microlipidotus, which is a member of a mainly Australasian group. According to WCH Clinical Toxicology Resources, this snake has an untreated lethality rate of over 80%, which is not good. If you're bit by an inland taipan, like, you need to get immediate medical attention, otherwise you're done. Like, that's just how these cases tend to go. We also find the coral snakes, and the sea snakes and the kraits. Now, the latter two here are some of the only marine reptiles in the world today. These species live most or all of their lives in the sea and are found across the Indian and Pacific Oceans, where they feed on eels and other small fishes. What is remarkable about the marine snakes is that they do not form a single clade that colonize the oceans. Instead, the sea snakes and the kraits belong to 2 unrelated lineages with their own distinct characters. So, for example, sea snakes give birth to live young, while kraits are egg layers. And of course, the other members of the Elapoidea include the clade Lamprophidae, or the African house snakes and their relatives, one of which is shown here at the bottom right. In fact, the majority of the other elapoid groups are mainly African and Southwest Asian in distribution, with only a few groups ranging into Mediterranean Europe and as far as Central Eurasia and southern China. And this has prompted earlier researchers to propose that Elapoidea itself must have had an African origin, given that so many clades are found there. Thus, the elapids and other non-African groups must have dispersed from there. And this is known as the out of Africa hypothesis for snakes. Not unlike the out of Africa hypothesis for humans. You can see my lecture series for more on that. However, there is serious phylogenetic evidence lacking to test this hypothesis, particularly a phylogeny that is time calibrated and includes samples from all of the subclades, not just a few. Winnell and colleagues sought to do just that. They sampled 65 species of elapoid snakes and their close colubroid relatives, which was then added to a previously compiled dataset that included 434 species. They utilized available fossils of snakes to calibrate the various clades, as well as tested for factors like past hybridizations between species, incomplete lineage sorting, for example. And all of these were then given a broad geographic label within one of 6 categories, based upon prehistoric continental arrangements over the Cenozoic era, which is, of course, during the time that elapoids and colubroids had evolved and diversified. There's sort of a Cenozoic phenomenon, if you will. Now, plugged together in totality, the resulting tree can be seen here at the right. So the American clades are in green, the African and Arabian are in orange, Oceanian is in purple, European in yellow, Asian in black, and marine in blue. Right away, the primary color at the base of the elapoid lineage, over here at the top, is black, not orange. And that points to an origin of the group not in Africa, but in Asia, at the earliest 45 to 41 million years ago, which is contrary to the previous hypothesis. Africa still played a major role in elapoid evolution, but as a separate and later colonization event, around 37.5 to 24 million years ago. In fact, this African radiation was just one of 4 out of Asia dispersals that occurred during the Cenozoic. Africa's cobras, including the members of the genus Naja, entered the continent 23.9 to 12.5 million years ago. The genus Ellipsoidea, the ancestor of the African garter snakes, entered 25 million years ago. And mambas of the genus Dendroaspis entered Africa around 18.9 million years ago. And these dispersals are well within the time frame that the African and Arabian plates collided into the Eurasian plate during the Oligocene and Miocene epochs, which famously created the Mediterranean Sea and pushed up the Alps and the Iranian Plateau. In fact, these dispersals were not all one way either. We see that the genus Malpolon, the Montpellier snakes, migrated from Africa to Europe about 17 million years ago, at about the same time that the genus Samophis, the sand snakes, moved into Asia. And of course, only 2 million years later, cobras moved into Asia as well. And of course, there were several other dispersal events from Asia into the Pacific and the Americas that were reported. So it seems that mainland Asia, at least, was the hub from which the elapoids slithered across the world. This is especially confirmed by the phylogenetic sequence that we have here, which is now more complete, which showed that the 2 earliest diverging lineages of Elapoidea are all Asian groups, a clade Cyclocoridae of the Philippines and a genus Samodynastes of Indonesia. And from there, we see a major split between the other elapoids, and then one large clade dubbed the Afro-Malagasy group, which became almost completely Afrotropical, as we've just seen. Now, the fact that this was not fully detected seemed to be a result from a lack of fossils of elapoids from the right time frame during the Paleogene period, as well as general assumptions about prehistoric species ranges and dispersals based upon data from living taxa, which we've covered numerous times on this show before. As the team discovered, having data from a larger range of species and having more fossil calibrations added to a phylogeny can make all the difference in uncovering the histories of dispersal for living organisms. And so it's kind of neat to see this sort of reversal in snake evolution and just kind of see the clarification here of just how wildly diverse we understand the colubroids and elapoid snakes, in the 21st century. And so I definitely wanted to share that with y'all. Albert, do you have any comments?
Yeah, this is fascinating. I mean, as always, I always like this type of kind of large scale macroevolutionary analysis that deals with phylogeny and biogeographic history and major evolutionary trends and patterns. And it's certainly a very fascinating group of animals to do it on. The elapoids are certainly some of the, or at least they include some of the best known and in some cases, feared species of snakes, and not entirely without reason, because some of them can be potentially dangerous to humans. In fact, quite a fascinating idea I've seen is that things like the spitting cobras that are able to kind of actually shoot their venom at aggressors, which I use as a defense, might have co-evolved with primates like us, basically, because primates don't like snakes very much, and primates are, quite can be quite dangerous ourselves. We can throw things and kill snakes that way, or use tools to kill them. And so it has been suggested actually that spitting cobras kind of evolved the ability to spit as a basically anti-primate defense. And so that's quite a fascinating idea to think about, that we have this kind of intricate history with these snakes. Of course, nowadays, it is reasonably straightforward for most people in the modern day to avoid conflict with these snakes, and so we can generally admire and respect them from a safe distance.
Absolutely. And I'll definitely give one more comment here before we go to the end credits. If anybody here has like kind of second thoughts, maybe negative thoughts about zoological parks, consider that zoos play a very important role in providing anti-venom to people who've been bit by snakes
Yep.
around the world. Many zoos keep these species in captivity and are able to, extract their venom and create anti-venoms from them. And so often what will happen is if somebody is bit by a snake or there's an accident at another zoo, like all you have to do is just call up the nearest zoo and say, "Hey, do you have this antivenom for this species?" "Yep, we have this species. Here you go." And usually it's like free of charge, this service. So I think that's just another, fascinating way that we've been able to help people around the world with snake bites through the use of zoological parks. So definitely give a shout out to all you zoos out there who are, helping people not die
Yeah.
or seriously not get injured from these snakes. Because
Right.
yeah, so some of the stories and accounts that I've read about, you know, different species and like the level of venom that they can puncture. Some species had said that they can kill 100 mice with like one bite, one bite dose of venom
Right.
um, at the fullest extent, and it's just that's scary to think about. But it's important to note that venom, this is not like a negative thing. Like snakes require venom to help kill their prey, which is otherwise can be large and dangerous and have nasty biting parts. And one of the
Yep.
best ways to dispatch prey is to knock them out, kill them first before they can get you. And the level of venom that a snake has tends to be in proportion to the types of prey that it eats.
Yep.
Um, which goes to show why some snakes are so venomous. It just so happens that that level of venom can correlate to like a single human body and can cause really nasty effects. Um, so yeah, there's a lot of myths and kind of negative associations with snakes like these, but I think it's important to note that like they have their roles in the ecosystem, and that just because a snake is venomous doesn't mean it's any less important than a non-venomous snake. And we should really do our best to protect all the snakes out there because they have a right to exist and they play roles in their ecosystems.
Yeah.
And
I mean, and related to that is that all snakes with a medically significant venom, they're not really interested in picking fights with humans because, as you note, like the main function of their venom is to take down their prey, but we are way too big for any of these snakes to eat. So the only reason that they would have to fight a human is if the human was, or they thought the human was directly threatening their life. And even then, in the vast majority of cases, the venom is a very last ditch type of defense because first of all it's a waste to use it because it takes a lot of energy for them to produce, and for another, if it's a fight to the death, like a human is going to beat one of these snakes every time, right? Like even if it manages to bite you and fatally like inject its venom into you, like you're still going to be able to kill it first before the venom gets to you. So there's definitely a lot of unfair characterization of snakes, especially dangerous ones, and it's good for snakes and good for people for us to be able to better understand what they are as organisms.
absolutely, absolutely. Well, I think with that, that is the end of our news episodes. I want to thank you all so much for joining us today and listening to these fascinating stories about so many diverse organisms on the planet. But I think now it's time to move to our general acknowledgments and end credits. So, of course, we are on Patreon. That is patreon.com/time and clades. Patreon is a special website where you can send monetary donations and get exclusive access to behind the scenes content. In our case, if you pledge any certain amount, you can get updates on upcoming news stories that we're going to cover, see special projects that we have planned in the pipeline, and of course, we have a different tier system as well that has many perks as well. So, for example, like you can get shout outs at the end of each episode. It's like the following people are owed. Of course, I want to thank my sister Julie and our friends Paul, Denver, Tristan, Frankish, and Valdinunzio for their contributions to this series. Every little bit helps, and we appreciate everything that we get. And all your donations will help us continue our series and develop new projects and expansions. And of course, moving to our general acknowledgments, I'd like to thank our good friends Henry and Alicia for their contributions to this series. Henry, of course, is responsible for the wonderful theme music that opens each episode, and Alicia is responsible for the color scheme of Albert's Alvarezor avatar, cheery little fellow. Of course, moving on to the next slide, just our general where to find us. Of course, we are on Twitter. We are at Time and Clades, where we usually upload episodes as they are premiered, but most likely you're watching us through our YouTube page, through Time and Clades. So if you wish to like and subscribe, that's always appreciated. If you have any questions at all about the topics that we've covered today, or just anything in general, you have three ways to reach us. You can send us an email, timeandclades@gmail.com, or you can tweet at us on our page, or you can send a YouTube comment, and we'll respond in kind. And of course, if you wish to read any of the papers that we have today, or any of the other things that we've mentioned as well, go to our description for links to references, where we have everything listed there for you. Now, a couple announcements that we've been teasing that we want to make. Um, First and foremost, we're going to be taking a slight hiatus from the show. Um, I know we've been trying to keep up with a monthly or at least bi-monthly schedule, but we're going to be pretty preoccupied this October. Albert, do you want to reveal why that is?
Well, it turns out that we are going to be attending the Society of Vertebrate Paleontology conference in Minneapolis this year. So I've been going to Society of Vertebrate Paleontology, or SVP for short, conferences for quite a few years now, but I believe this will be your first one. So first one where both of us are going to be present. It'll be a great time, and we will tell you all about it when we get back. But in the meantime, we will be taking a short break from the show for a little bit.
Right. It takes us some time to prepare for these episodes, and we definitely wanted to make sure that we at least had something out for y'all in the meantime. But of course, nothing says that classics can't be revisited. Of course, we have two complete lecture series now, and the update specials associated with those: Dinosaurs, the Second Chapter, and Humanity, a Prologue. And of course, the first two episodes of Humanity, a Prologue Season 2 are already out. So if you want to, learn some information about like the history of anthropology or the evolution of the human lineage from the origins of life to our last common ancestor, those are available, as well as all of our subsequent news episodes and reviews and interviews. And not to fret, even though we'll be gone for a little bit, we still have more surprises in store. So we've just been given permission to announce this. My partner Alari, who is a very prolific traditional artist, especially with paleo art, he is going to be heading a new lecture series for our channel. That's right. So he's going to be covering basically the history and practice of paleo art. Now, what is paleo art? Well, if you've ever visited a museum or seen like a technical book or have seen a documentary series, the individuals who use all of our current fossil evidence, anatomical studies, geographic evidence, whatever that we've learned about prehistoric times, then take that information and create reconstructions. So it's basically like the rigorous science-based approach to creating restorations of long lost fossil organisms across all geologic timescales. And so a lot of questions go into that. Well, how is it done? How is the evidence used? How exactly, can you map muscles and skin and flesh onto a skeleton, or much less like a shell? How can you reconstruct environments? when there's little fossil evidence, how do you get like a toe bone? You get a whole animal from that. Well, not to fret, Alari is going to be there to explain all of that and more to all of y'all. Um, we do not have specific release dates yet for this lecture series, but it will be forthcoming. And we are both very excited to have somebody new on
Hmm.
to share their expertise with the world. And we hope that you all will be there to join us when that happens. And we'll keep you all updated, of course, as it goes. Um, but I'm certainly very excited about that. I'm sure you are as well, my friend.
Indeed.
Absolutely. But till then, we will see y'all later. Take care. Have a wonderful rest of your day, and we'll see you next time.
Yep. See you soon.